Good afternoon, guys. It's me, Miss Cherry. I'm doing this over video because you guys said that the Zoom was rubbish or whatever. I'm not offended at all. I'm going to blame Virgin Media for it. We're going to see how this goes. I should be in by Friday, fingers crossed, track and trace notwithstanding. So this is just me going through this PowerPoint. I will tell you at various points extra information you need to know as we do in the lesson. I'll also tell you when you need to stop and you need to do some work by yourself. Okay, so I will put on Edmodo that you need to download these videos before you do your lessons and before you get to class tomorrow. So hopefully when I come back and see you on Friday, we're all up to speed and we are confident and happy with all of the content. On Friday, you will have the opportunity to go back over things with me, things that you didn't understand, maybe you weren't too sure about an organelle. But for now, I'm going to assume, because I can't see you, that we are all okay and up to speed. And we're going to get started today by looking at mitosis. Okay, so again, I'm just going to keep talking. I'm going to tell you when to stop and then I'll have a little pause. And then I'll start going through the answers, okay? So for now, this is your starter. Have a go at these questions. I assume that hopefully you'll be able to take 5 minutes to have a look at these. And you should start now. So pause the video, spend a few minutes answering these questions, and then play again and I go through the answers. Okay, so for number 1, how do eukaryotic cells divide? Now hopefully, knowing what you do at GCSE, well this is a good start already, isn't it? Knowing what you do know about GCSE, you should have been able to tell me that eukaryotic cells divide by mitosis. Okay, that's the production of 2 genetically identical pause for talking, 2 genetically identical daughter cells. Okay, so the genetically identical daughter cells means that effectively, mutations aside, these cells should be clones of those eukaryotic cells that were made or that they came from, that parent cell. The replication of sex cells, which we can also call gametes because as we know, we are smart, is known as meiosis. Again, this is going back to GCSE. Mitosis happens in mitoses. Meiosis happens in myovaries. Okay, if you don't have ovaries, it happens in the testes, but that doesn't have too much of a ring to it. Now, genetic material in bacteria appears in one of 2 forms. More often than not, they occur in the cell at the same time as well. You can have a nucleoid, which is sometimes known as a chromosome. And this is normally the biggest part of DNA. So it looks just like a big squiggle. Okay. Alternatively, you can have plasmids. And these are generally numerous. So there could be one or lots of them in the cells. And remember, they are able to replicate independently of the cell itself. Independently. Okay, so they're short circular bits of DNA. That's really important. That's going to be super duper important when we get to topic 8 and we start looking at gene technologies, because these plasmids can be opened up and spliced and cut apart and stuff to see the stuff inside and to start manipulating that genome. Viral replication is done using a host. And as I said, on Friday we're going to take a look at HIV later in this module. And the need for cells to divide is obviously your standard GCSE answer of growth and repair. You guys are not the same size as you were when you were born because you've grown. You've grown because you've accumulated more cells through the process of mitosis. And that's what this lesson is focusing on today. So we're going to have a look in greater detail at what mitosis is. We're going to identify where DNA replication takes place, and this is going to take us into lesson 2.9. We're going to skip 2.8 because that's a required practical, but it is based on this lesson. Okay, so next week after your required practical, you will be next week after your assessment, you will be undertaking that required practical. As I told you last week, we've got 3 required practicals to do before Christmas now, so it's going to be busy, busy, busy. We're going to be having a look at when DNA replication takes place, and again, feeding into lesson 2.9, we're going to see what happens when this goes wrong. And finally, we're going to see why mitosis is important. Okay, so let's have a quick recap of GCSE and the things that we should hopefully know. Okay, so mitosis and meiosis are our 2 different types of cell division. We know that mitosis produces the 2 genetically identical daughter cells that we spoke about in the starter. So we've got our first cell and it splits down into 2. This is going to occur in my body cells, as we said. It's occurring in mitoses. Meiosis, on the other hand, produces genetically distinct daughter cells. Okay, so they've had their genes changed. And these have got half the number of chromosomes that a normal cell should have. Remember, the key word for that is haploid. Okay, half the number. Conversely, going back to the mitosis session, we say that these have got the number of chromosomes as a parent cell. This is going to be a body cell, so we say that mitosis produces diploid cells. Meiosis occurs in our sex cells, which we're now going to call gametes. And, hopefully this is just a revisiting of what you've done previously. Now, cell division is sort of an unspoken key thing to do with life. So the idea of growth and progression of life is that your cells are able to keep dividing, and that to grow, you effectively produce more cells produce cells faster than the rate at which they die. Okay, we apparently the scientific definition of death is when your cells start dying quicker than they're replenished. I think it's about 26, 27, so I'm on the downhill slope from here, but you guys have got a few years left. But cell division, aside from death, is really, really important for single-celled organisms. Remember, we can say single-celled as unicellular, and this is things like yeast, and they just asexually reproduce by dividing like you see in the picture. For us, the big multicellular organisms, the more complex things, plants, animals, humans, this cell division process enables growth and replacement or repair of damaged cells. And these cells could be damaged from going to the gym and pumping too much iron, so you start tearing your muscle cells. This could also be because you've had an infection and it started to damage maybe some tissues in your body. There's lots of reasons why your cells might need repairing. And remember that cell death is just a standard part of the life cycle of a cell. It's not a bad thing, necessarily, for cells to die. Now, chromosomes are key, pivotal in cell division, and actually it's the movement of these chromosomes that tell us which stage of mitosis our cell is in. Okay, so normally chromosomes just look an absolute mess in cells. You can't really see them because they're just long, thin, wiggly strands. And you can see here in this really poorly outlined diagram of a chromosome that actually inside of it, it's made of these lots of little fibres. Okay, now this little fibre, as you can see further in the diagram, is called chromatin. Okay, chromatin is made up of DNA, the green strand, wound around proteins called histones. Now this means that our DNA, if it's wound up, is not as long as it could be if it was left to stretch out. Okay, that's good because it means that it can compact nicely into our cells. The arrangement of chromatin in the chromosomes is in 2 arms. Okay, each one of these arms is called a chromatid. So I've got one chromatid there and one chromatid there. They're not technically X-shaped. They're not actually made of 4 points joined together with a ball. Okay, they just touch at a single point, and that's called the centromere. Okay, so the chromatids are the arms. The centromere is the middle. Okay, that meeting point. Now, before we get onto the stages of mitosis, it's really, really important that you guys recognise that each chromatid is identical. So if we say that we've got one arm of a chromosome here, and we've got the second chromosome there, okay, these might all be chromosome 2 from your mum and chromosome 2 from your dad. Okay, regardless of who they came from, if they carry specific genes, so maybe your mum carries genes for blue eyes and your dad carries genes for green eyes, that gene is going to be located at the same point, regardless of whether it's for green or blue eyes. Okay, likewise, you're going to have some chromosomes, and I'm sorry if you can't see the green, I'll change it before you guys shout at me over email. Hopefully this darker green is a little bit better. There'll also be more genes for things like enzyme formation. Okay, but notice that even if, like I said, you get blue eyes from the blue eye gene from your mum and the green eye gene from your dad, the genes found on each chromatid are identical. That's really, really important. If you take nothing away from this slide, each chromatid on a chromosome is identical in the genes it carries. Okay, these genes were identified through things like the Genome Project, but what it means is that if I cleave a cell down the middle, okay, whatever genes are found on this chromosome and go to the cell on the left, and whatever chromosomes are found on the right hand arm and go to the right cell, all that means is that those daughter cells that I've produced from that division are going to have the same genes. Okay. Now, the way that I get that cleavage is through mitosis, and this is what the rest of the lesson is going to focus on. You will have seen on Edmodo, hopefully, if I remember, with this video, I've uploaded a little picture cycle thing. If you wanted to, before you go to the lesson, you can print that off. If not, you can spend a little bit of time in a second drawing those pictures out. It is quite important to be able to recognise them in a cartoon. I'll show you what I mean in a second though. Let's not get ahead of ourselves. So this is the diagram that I was telling you about. If you've printed it out, that's fine. You might want to make it so it's quite small in the middle of A3 paper. You can draw them out if you're still not very happy with them. Alternatively, you can completely ignore this picture because there's going to be, I think, slightly better cartoons on the next few slides. Okay, so this shows you the process of mitosis. Now, what we can see here is an ongoing cycle. It doesn't tell us too much detail, though, about the time spent in each phase and what happens. Now, this is because the time spent in each of the stages differs depending on the cell type that you've got. Okay, and when we get to the cell cycle, we'll go through this in a little bit more detail. So we're going to start up here. Okay, we're going to start by looking at interphase. So if you haven't got the diagram in front of you, that's fine, but if you want a little subheading, interphase is where we're going to begin with. Now, interphase is what happens before mitosis. So if you look quickly into the top right of this screen, if you imagine the cell cycle like a pizza, okay, it constantly goes round and round and round, mitosis makes up a very small part of that cell cycle. Okay, interphase is the bit before it. Okay, I can't write phase because of that stupid button up there. But in interphase, we get genetic material replicating. You can also say duplicating or doubling. Don't say, oh, the genetic material increases, because that's just a little bit arbitrary and a bit random. So what that means is that our chromatids, remember the arms, go from 46 to 92. And this is how we get, when we used to do GCSEs, when you did your GCSEs, not that you did, you would have said that the genetic material increases from 46 to 92 chromosomes, and then it splits down again into 46. Okay, our two daughter cells have 46 chromosomes each. Now, interphase is just this bit. You do not need to know any further detail about what happens in interphase. That's it. Done diddly, genetic material replicates. DNA replicates, chromosomes replicate, whatever, done. We're moving on now to the stage called prophase. Okay, and this is split into two different stages, early prophase and late prophase. Now this is where the diagrams get a little bit better. Okay, I prefer these. I think they show the process a little bit more visibly. If you want to, in your notes, maybe leave some spaces for these pictures, and you can go and find the PowerPoint on Edmodo and download them and stick them in. Up to you, don't really care. Now, early prophase is the very first step of the mitosis process. Okay, so we've done our interphase, we've duplicated our genetic material. Now what we're going to do is we're going to take those chromosomes, which were initially very long, very thin, very fat, it's not fat at all, very skinny and a bit wishy-washy, and we're going to condense them. Okay, so we're going to take them from the long, thin structures. Oh, case issues, hold on, bear with. We're going to take them from the long, thin structures, and we're going to make them shorter and fatter. Now, what this means is that down a microscope, they become visible. Okay, so the first stage of mitosis is identified by actually being able to see those chromosomes. In our cells as well, we can see that the centrioles, which are those cell organelles that you should have noticed or learnt about last week, they're lots of tubes in a little circle, they move to the poles. Okay, now if we're thinking about the world, we normally consider the poles to be the north and the south. When we're drawing cells, it's traditional to make sure that the poles are on the left and the right of the cell, not the top and the bottom. It's not really going to be wrong if you do it at the top and the bottom, but you know, it's a little bit easier when we come to the next few stages, drawing them sideways. At the same time, so we've got chromosomes condensing, centrioles moving to the poles or the side of the cell. We've also got the nucleolus beginning to shrink. Okay, so initially we would have had a small, more dense region inside the cell, or inside the nucleus, sorry. But over time, through this early prophase, that starts to disappear. The next step is late prophase, and we're going to look at that now. Now, late prophase is what happens after our chromosomes condense. Okay, so now we can see the chromosomes as two fat chromatid arms, and these centrioles at the side, okay, we'll draw the centrioles and the arrow in, start to produce something called spindle fibres. And these are effectively, like if you imagine an octopus, the spindle fibres, are all of the arms. Okay, so we've got spindle fibres, and they are made by the centriole, which would be the octopus's head, for example. Okay, this is what happens in animal cells. In plant cells, the centriole isn't really a thing. Okay, instead they have something called the spindle apparatus, which does exactly the same job at producing those spindle fibres. Okay, now as you can see, the spindle fibres, I'm just going to put SF, start to reach towards, reach towards the chromosomes. This is going to be super important for our next step. At the same time, you can see that this picture has got a very faint nuclear envelope. The nuclear envelope has to disintegrate, otherwise our spindle fibres cannot reach the chromosomes to pull them apart. Okay, so the nuclear envelope is disintegrating. In early prophase, we had the nucleolus disintegrating, and now we've got that nuclear envelope going away too. That is the end of prophase. So if you want to spend a little bit of time now, pause me before we move on and summarise the stages of early and late prophase. From late prophase, metaphase follows prophase. So far we've had prophase, and then we've got metaphase. In this stage, we start to see the movement of our chromosomes. Okay, so these spindle fibres have come from the centrioles and they start to attach themselves at the centromeres. Okay, remember the centromeres are the points where our chromatid arms meet. What am I talking about? You can also see that if I flick back quickly to metaphase, the chromosomes have started to line up at the centre of the cell. Okay, this is called the equator. Now what we've got here is we've got spindle fibres. Okay, I'm going to highlight these, hopefully in a colour that you can see. Spindle fibres in blue, joining to the centromeres, which I've just highlighted in black, but I will draw a black dot. Okay, and these centromeres and spindle fibres mean that each chromosome is attached to both poles. Okay, it's got 2 points of contact. So this chromosome here that I've put a little star next to at the very top is attached to this centriole and this centriole. Okay, now in terms of moving our cells, or moving our chromosomes, that's really important because it means that the even drag, once these spindle fibres contract, is going to pull each of those chromatid arms to the poles. And that's the end of metaphase. Now, on the next slide, there is a little bit of a caveat. It's more of extra information that you might want to know about, especially if you're chasing an A or an A star. Okay, and this is called the metaphase checkpoint. Now, the metaphase checkpoint is exactly what it says on the tin. Okay, metaphase has happened. You've got your chromosomes lining up. I'll draw it here. You've got your chromosomes lining up at the centre of the cell. You've got your centrioles, and then you've got some spindle fibres, which I'm just going to draw in dashed lines, attaching each of those chromatids to the poles. Okay. Now, if I was a cell and I was in a little bit of a rush, I'd be like, okay, that's fine, yeah, let's go. Okay, yeah, that's fine, let's go. Let's keep dividing. But instead, the cell recognises that if this bit is wrong, if the metaphase stage hasn't happened correctly, then my cell is not necessarily going to divide perfectly. If the cell doesn't divide perfectly, then I could get some mutated cells, or I could get some cells that are not suitable or not able to survive. Maybe they've got too many chromosomes or too few. Okay, now this whole process of checking over the cell is called a checkpoint. And smartly, scientists have decided that as this happens in metaphase, we're going to call it the metaphase checkpoint. Now, at the metaphase checkpoint, the cell is checking that the chromosomes are attached to both spindle fibres. That are connected to both centrioles at either side of the cell. Okay, if the cell is not connected to spindle fibres at the centromere that are connected to both poles, then we have something called cell arrest. Okay, the scientific word for cell arrest is apoptosis, and that is a stage of programmed cell death. Now what this means, cell death. What this means is that if you have a look in the diagram quickly, if my chromosome is only connected at one side or from one spindle fibre, the body sort of goes, you're going to cause me more hassle than it needs to be. I'm just going to kill you. I'd rather make a new cell. Okay, and that's what it does. If it recognises that the division of that cell is going to be problematic, it just gets rid of it. If you want to use it in a sentence, you say the cell undergoes apoptosis. You can't really say things like it apoptosizes. It doesn't really make much sense. Okay, I'm going to leave it here. We're going to move on to the third stage. So we've done prophase, we've done metaphase. The next step is anaphase. Okay, so as we said, we are prophase, metaphase, and then anaphase. Now here, you can see the cell has very, very visibly changed. We've got these spindle fibres that were initially all in the middle, attached to the centromere, as we drew in the last one. Okay, they have now contracted. They're contracting, remember, it's an active process using ATP. Okay, and remember, if you want to have a little recap, try drawing the structure of ATP now. Now what we've got is that contraction of the spindle fibres pulls the chromatids, those arms, to the opposite ends of the cell. Now I've made a bit of a boo-boo. Throughout this whole talking process, I've been referring to each of those bits of the cells, the chromatids, as the chromosomes. That was incorrect of me. Okay, when they're joined together, each of the arms are called chromatids. Okay, tid, I'm just going to put tid. When they're separated, as they are in anaphase, you can now call them chromosomes. Okay. So, in actual fact, I should probably go back and change all of this video, but I've spent about 15 minutes doing this so far. I'm not going to do that. That's me telling you that I am wrong now, though, okay? So make sure, if you've been calling it chromosomes throughout the whole of it in your notes, go back, make sure you're changing them to chromatids, and only after anaphase can you call them chromosomes. So those chromosomes are being dragged to the opposite ends of the cell. We can say that they're dragged to the opposite poles as well. That's also really good because we're using our key vocabulary there. Now, the final thing that we need to be aware of with anaphase, and this is going to link to things like our specialised cells, we know that it's an active process, the contraction of those spindle fibres, and that's going to come from ATP produced by the mitochondria in processes of aerobic respiration. Okay, now what we've got here is we've got a massive collection of those mitochondria gathering around the spindle fibres. Okay, so these cells that undergo a lot of mitosis will be really energy heavy, but also produce a lot of energy as well. Okay, so these are very, very metabolic cells. And this is going to be important when we come on to lesson 2.9, because we're going to have a quick look at cancer. Okay, so that's just a warning in advance if any of you are affected, and I'll say it again at the beginning of the next lesson. If any of you are particularly affected by cancer or you've got things going on in your family, just know that it's the scientific basis of cancer. It's not looking at any sort of majorly emotional aspects of it. Yeah, but I'll get to that when we get to there. Okay, now we've got a little task for you to do. So I'm going to stop this slide, moving on to the next one. This is a you task now. So what happens if the spindle is treated with chemicals causing it to disintegrate rather than contract? What's going to happen to the cell? Okay, so again, if the spindle is treated with chemicals causing it to disintegrate rather than contract, what happens to the cell? So I'm going to pause now. You can pause me too. Spend a couple of minutes thinking about it and then play me again and I'll go through the answer. Okay, so if our spindle is disintegrated rather than contracting, that means that the centromeres of the chromatid. Remember, we're using the word tid. Right, so the centromeres of the chromatid arms are not connected to the spindle fibres. This means that the spindle fibres can't contract because they've disintegrated. So the chromosomes, okay, because we can call them chromosomes once they've separated, won't move to the opposite poles. And in turn, our cell won't divide. Okay, because even though we haven't finished learning about mitosis, or we haven't finished the steps, divide. Even though we haven't finished the steps of mitosis, we can recognise that if the chromosomes are not being pulled to opposite poles, there's going to be no cell division. Okay, if the cell won't divide and it's sort of gone a little bit blah, then we can say that the cell undergoes apoptosis. Okay, remember we're using that word. If you can't remember apoptosis, programmed cell death is also fine, but that's the best word to use. Okay, so if that's 1 mark, that's 2 marks, and that's 3 marks, give yourself a score out of 3. We're going to move on now to the final stage of mitosis. Now, the final stage of mitosis, after we've had prophase, metaphase, anaphase, and now we are at telophase. Okay, so again, you can see that the structure of the cell has massively changed. Okay, we've reached the poles of the cell now. So we've gone towards those centrioles, and we've got separate chromosomes in each half of our original cell. But now, those chromosomes start to uncoil. So remember in prophase, it was really important that they condensed. Now they start to relax a little bit. They become less distinct. So we can't see them as well. Less visible. Down a microscope, they look a little bit more fluffy. And I'll show you some microscope pictures in a second, and it'll make a little bit more sense. Now in telophase, or telophase, whichever way you want to say it, we can start to see the nuclear envelope reforming. And you can see on this diagram that it's very, very faint on the outline. Very, very fuzzy. But we're starting to get 2 distinct nuclei produced. We're very nearly at the stage that we've got 2 daughter cells. The final thing that needs to happen, which isn't technically part of mitosis, because mitosis is relating specifically to the division of the nucleus, okay, and now we've got 2 separate nuclei here, something called cytokinesis. Now this is literally the division of cells. Okay, so if I make a quick note up here, cytokinesis is the division of cells, whereas mitosis relates exclusively to nuclear division. Okay, now in animal cells, all that happens is that if you imagine like your teeth when you bite a mushroom, a marshmallow, that your teeth are doing a pinching motion on the marshmallow. And that's exactly what the same that happens in animal cells. So the edge of the membrane pinches in and produces these 2 little divided cells. In plant cells, however, there's not a picture on the next slide, but if we've got 2 distinct nuclei like this, the plant cell actually lays down a new cell wall in the middle before the 2 cells separate. Okay, and that process of laying down a new cell wall comes before the separation process, which, you know, for some reason you need to know it's called cleavage. Okay, it's just that division. And that's it. That's the process of mitosis. So the rest of this lesson is going to be looking at how we see mitosis under a microscope. That's relating to our required practical, and then taking a look at how other organisms replicate. At the end, I will shut up for a bit because we've got some exam questions. Okay, so moving on to how this looks down a microscope, you can see here that it's very difficult if you are unsure of what you're looking at. Okay, obviously, because otherwise everyone would do it. So let's just go through these, and we're going to make a few notes on what we can see down a microscope. So to begin with, we start at interphase, and we can see two distinct nuclei. Okay, we know that from interphase, we're going from 46 chromosomes to 92. Okay, we've duplicated our genetic material, doubled it up. Now, if we move around to prophase, notice here that it's very hard to distinguish between early and late prophase. In the next task I give you, I am going to ask you to distinguish between them because I'm horrible, but for this picture, for a little bit of reference, you don't need to know. Now, we can tell this is prophase because our chromosomes are more visible. Okay, you can see that the cell has become very much more dense. Chromosomes visible. Okay. Now I've made a boo-boo again. Chromatids are visible. Okay, remember now they've doubled up. So we've got the arms, we've got the chromatid arms joined at that centromere. So chromatids are visible, and notice as well that the edges of the nucleus have sort of disappeared. So if you look back at the image for interphase, the nuclei are very distinct circular structures, whereas here it's a little bit more of an arbitrary blob shape. The nuclear envelope has disintegrated. Now metaphase, very, very easily visible. Our chromatids, I'm using the right word this time, chromatids line up at equator. And also, if you look very closely, and I'm going to try and zoom in without ruining this whole thing, you can sort of see, around here and around here, the spindle fibres. Okay, so zooming back out again. Yes, well done me. You can sort of see the spindle fibres there. And they're going to be attaching to the centrioles. Now, if I carry on round to anaphase, okay, PMAT, so prophase, metaphase, anaphase. Ooh, zooming in again, you can see that we've got some form of drag. All right, so our chromatids are going to be looking from this kind of shape, the X, and then they're going to be pulled. So they're going to start looking like when you were a child drawing little birds. Okay, you can see they start to drag. So our chromosomes now being dragged. Now notice you can still see them as individual lines. They're very, very dense, very, very visible. And then we move finally on to telophase. Now here again, it's very difficult sometimes to distinguish between anaphase and telophase, because really this diagram for telophase, I'm going to put that as step 5, very similar to the anaphase picture. It might well have just been dragging slightly further down the line. Okay, but the distinction here is that all of the chromosomes are tightly packed together now. Okay, so these are effectively our distinct nuclei ready to be reformed. Okay, so hopefully I'm not going to cock this up. I'm going to stop recording my voice for a second and change the slide, and then I'll be back. Okay, so make sure you're taking a look at this, and we'll go again in a second. Okay guys, me again. Hopefully this hasn't paused and ruined the rest of my video, fingers crossed not. Using the previous slide, can you please spend a bit of time now trying to figure out which stage of mitosis each letter shows? Once you've done that, try and put the stages in order. Now this might be a little bit tricky because the picture is frankly abysmal. I apologise, but you know, can't help what you can't help. The first thing I would recommend doing though is blocking off your cells. So I'm going to do that in, what colour is going to be best? I'm going to do that in a really nice hot pink, just so it's super visible against the horrible greeny blue background. So if we're looking at A, I'm looking very closely at the edges. Okay, you will notice that it looks like there's a little division in the middle, but you know, that's meant to be there. That's good. So if I can see two distinct nuclei where cell A is, and I can also see a slightly, and I'm going to rub that little pink line in the middle out again so you can see it a bit clearer, a very faint, I don't know why I'm whispering, very faint line, I know that my cell at that stage must be undergoing cytokinesis. Okay, so I've done A for you. Spend a little bit of time, I'm going to shut up again for a second. Spend a little bit of time trying to figure out what B, C, D, E, F, G and H are, and then see if you can put them in order. So you need to pause me now, and then I'll be back in a second, or play me again in a second, and we'll go through the answers. Pause me now. Hello again. Hopefully you enjoyed that game of horrible cell images. Going through the answers then. A was cytokinesis, we said that together. B is late prophase. Now we know this is late prophase because if we look very closely, I'm going to try and zoom in again and not ruin it, we can see the chromosomes, but we can also see, so we can see some of the bigger chromosomes in the wiggly structures. We can also see that if we compare the edge of where the nucleus should be, so all around here, to literally the cell just below it here, the nuclear envelope has effectively disappeared. The edges are a lot more fuzzy than they should otherwise be. Okay, now looking at C, which is over to the left-hand side a little bit, you should have been looking at early prophase. Now here, in early prophase, if I just get rid of all of that rubbish, we've got our chromosomes condensing, but we can also still see the nucleolus. Now, the nucleolus is still very dense, so our cell outline is there. Our nucleus, blimey, I was doing so well. The nuclear envelope is still there. Go away, I don't know what I'm doing. Nuclear envelope is still there. The chromosomes are starting to become visible, but the nucleolus, that really dense part in the nucleus, still visible. Okay, it is starting to go. You can tell that from the fact that it's not as dense. Again, let me get my little rubber out and see if I can get that bit away. Go away. No, it's not working. Hold on, bear with. There we go. So it's a little bit more fuzzy around the edges though. All right, and that's fine. If we carry on to D, down at the bottom, we've got a slightly draggy motion. We've got anaphase happening. And in E, we've also got anaphase happening. So remember we said in the previous slide that anaphase can happen at various, or the picture can be taken at various stages of anaphase. But in both of those, D and E, we've got a kind of dragging effect. For F, you should have had metaphase because we can see that they're nice and at the middle. All of those chromosomes or the chromatid arms are lined up at the equator. G is where it starts getting a little bit interesting. So G was a bit of a trick one. Okay, G actually showed interphase, which isn't technically a stage of mitosis. Now finally, H is telophase, because again we said, remember, that we can identify telophase when all of the chromatid arms are touching one singular point. Okay, so we're starting to get that nucleus reforming. Now, if you'd been super smart, because you guys are, hopefully you would have got all of those right. If not, pause me now, spend some time going over those images. Also spend some time figuring out what order they should have gone in. And if you have, carry on. And your answer should have been G, interphase, followed by C, early prophase, then B, late prophase, F, metaphase, E, then D. Okay, although they're both anaphase, you can tell that E comes first because they're ever so slightly more lined up at the equator, whereas D, they've almost got that little gap between them. Okay, so E, D, and then H, where our telophase is, and then A, cytokinesis. Okay, well done to those of you if you got that right. If not, take another look, listen to my lovely dulcet tones again, and then you can move on. So that's the end of the part of the lesson about mitosis in animal cells. The next few bits of the lesson are looking at bacterial cells. So bacterial cells don't replicate or divide by mitosis. What they do instead is something called binary fission. Now, binary fission, I'm going to zoom out of this image a little bit, is basically mitosis in cells, but you need to know it as a separate process. I don't know. They are separate, I guess. So what we can see first is we can see our circular DNA replicating. Now, this is a rubbish diagram. I apologise. By circular DNA, what I really mean is the chromosome or the nucleoid. And from our original cell, I'm going to call this one where picture 1 is, the parent cell replicates, and both of those chromosomes should really be attaching to the membrane, but I've drawn little sticks on there, so that's fine. Okay. Also at the same time, we've got those plasmids replicating. Okay, so remember the first stage of mitosis, or before mitosis, we had interphase. In binary fission, the same thing sort of happens. So the genetic material duplicates. Once we've got that duplication of our genetic material, our DNA and stuff, the cell membrane begins to pinch. Exactly the same as what happened during cytokinesis. So you can see here, if I draw a little arrow, that the membrane is starting to work its way inwards. Okay, so it's not being sliced inside out, it works outside in. Now remember, our bacterial cells have got not just cell membranes, they've got cell walls. So these cell walls are going to grow, as you can see in picture 5, and they're going to cleave the two daughter cells apart. Now each one of those daughter cells is going to have one copy of the circular DNA, that nucleoid or the chromosome, but the key thing here is that they can have a variable number of plasmids, and this entirely depends on where the plasmids are in the cell at the time of cell division. So you can see if we follow the plasmids, I'm going to highlight them from picture 1 through to 5, these plasmids are free to move throughout the cell. Okay, so if at the stage of cleavage I've got all of my plasmids, sort of, if I go back to picture 4 and draw some more in green, if I've got them all in the bottom cell, then when my cell divides, I'm going to have a lot more plasmids in that bottom cell. That's absolutely fine. It just means that maybe that bacteria has a little bit more chance of mutation. That is A-okay. I'm going to go back. Could you spend a little bit of time drawing these pictures out for me and adding your own notes, making sure you've made some form of comment at the fact that the variable number of plasmids is okay and normal. And then we're on to the final steps of the lesson. Yay! Looking at viral replication. Now, before we move on to viral replication is a really big deal, given that we're in the midst of a global pandemic. Don't want to bring it up again, but that's why I'm not here. Now, in this image, we've got something called a bacteriophage. Okay. Now, phages are the science word for virus, and bacteriophages are literally viruses that infect bacteria. Sorry, I'll change from this green in a second. That infect bacteria. I'm not going to write the word bacteria because it's there. Right, back to that brown. Now, with this bacteriophage or this viral replication, the whole process involves the virus taking over the host. And it does this through the stages that you can see in the diagram. So we're just going to go through them. Again, we're going to spend some time at the end of this module looking at viral replication, specifically HIV. But if you can understand it from this slide, then perfect, will be in a great step for next time we come to it. So first thing we've got is we've got our phage, our virus, attaching to the surface of the host. And remember from last lesson, it's going to attach through attachment proteins. Those little bits on the outside that stick out a bit like the antigens on a cell, and they stick to a host. Then what we've got is penetration, which is a horrible way of saying that the phage basically stabs the host cell and incorporates or inserts its DNA into the host. Okay, so it inserts into host. Notice that the capsule and the envelope of the virus stay outside. Okay. Now that we've got our viral genome in the host cell, the virus incorporates its DNA into the host. Okay, so virus incorporates DNA into host. Now this means that when the cell would normally use the ribosomes to make proteins for the cell, once it gets to the bit of viral DNA, it goes, oh, okay, that's DNA, cool, we're just going to make that. And in turn, that means that the phage DNA becomes replicated, that viral DNA becomes replicated by the host. And that makes more copies of the proteins that are needed to build the virus. So over time, we're building up the little proteins, so the DNA, the capsule, all of the attachment proteins inside that host cell, that then during the maturation phase, are stuck together. Okay, so we call it maturation because these are early viruses here, and then they move on to being mature viruses capable of infecting things in the next image. Now finally, we've got basically like that scene from Alien. The cell lyses. Okay, that means that it breaks, and the newly made viral cells are released. Now the key thing about this is that when the cells lyse, so when cells lyse and viruses are released, they take part of the host membrane. Okay, now this is a bit like, host membrane. So this is a bit like if, imagine there was a whole group of Mr. Mojitas. Okay, if Mr. Mojita made up all of the population and I was infecting Mr. Mojita, I lived inside of him and I wanted to make more of me using his body parts. It's a bit weird, a bit disgusting. I hope he doesn't hear this. But once I break Mr. Mojita down, I can already tell this is a bad idea in my head. I'm going to go with it. So once I bust out of Mr. Mojita, I'm going to effectively steal a little bit of his skin and I'm going to use it as a little helmet. So all of the other Mr. Mojitas will be like, you can't see me, you can't see me, you can't see me. That's what these cells are doing. Or that's what these viruses are doing, sorry. So as they bust out of the host cell, they take a bit of the cell membrane with all of the proteins that that cell membrane's got on it, that the body or whatever organism recognises as itself. And in doing that, it's almost able to make like a little helmet to wrap around itself and hide from the immune system. Now that's us done in terms of the learning of the lesson. So I'm going to go, okay, I'm going to stop this recording. I'm going to spend some time, I'm just going to skip slowly through the next few slides. You can pause when you're ready to move on, or you can play when you're ready to move on and pause when you're not. So you're going to have a go at these questions. Is identical always good? So I would like you to evaluate the advantages and disadvantages of having genetically identical offspring or that are genetically identical to the parent. And you want to consider those things in purple at the bottom. So specialised cells, bacteria becoming resistant, and defence against diseases. Remember, biology is not just related to humans. So yeah, you might want to consider specialised cells in terms of humans, but we've got to think about prokaryotes. So looking at how this binary fission makes the identical cells in bacteria, and defence against diseases, I would like you to think about that in terms of plants, and specifically crops and farming. Okay, so spend a little bit of time thinking about those. Pause this, use your books, use your phones, use your iPads, use whatever, and then play again when you're ready. The next thing I'd like you to have a go at is the task recognising the stages of mitosis on page 79 of your blue book. If you haven't got a blue book, there should be one copy at the back of L9, unless someone's nicked it, in which case I'm going to find out who it is. And it's just the little green box at the bottom of the page. It's not too long, there's only 3 questions. You're just using pictures and recognising steps of mitosis. Once you've done that, in the blue book again, you can go to the back. Hold on, let me find the page. What number is this? 3.7. So if you go to page 610, answers 610, it's 3.7, recognising the stages of mitosis. If you struggled with the maths bit, go back to page 307 and just have a look again and see if you can figure out how they got to those answers for questions 2 and 3. If you're still struggling, make a note of it and come back and talk to me about it on Friday. Now the final few slides are exam questions. So again, I'm just going to skip the slide, you pause, and then the answers are at the end. Thanks very much for watching this, guys. I hope you found it useful. I will finish with the exam questions, but I'm going to stop talking now. Yeah, if you've got any questions, send me an email during your lesson time and I'll reply to them because literally got nothing else to do. Okay, I'll say bye at the end though. So exam questions. Scientists looking for treatment for cancer are investigating the use of substances called kinesin inhibitors, Ki. These inhibitors prevent successful mitosis. Some kinesin inhibitors cause the development of a monopolar spindle in mitosis. Figure 6 shows how chromosomes attach to a normal mitotic spindle and to a monopolar mitotic spindle. Suggest why the development of a monopolar mitotic spindle would prevent successful mitosis. So suggest, give reasons for, using your knowledge. You haven't heard of kinesin inhibitors, but look at that picture. Think about what we've done earlier in the lesson. Suggest why the development of a monopolar mitotic spindle would prevent successful mitosis. Scientists investigated the effect of different concentrations of a kinesin inhibitor, Ki, on mitosis of human bone cancer cells grown in a culture. Table 3 shows the scientists' results. So we've got concentration of kinesin inhibitor in nanomoles per dm cubed on the left-hand side and percentage of dividing human bone cancer cells showing a monopolar mitotic spindle on the right-hand side. A student who saw these results concluded that any future trials of this kinesin inhibitor with people, trials of this kinesin inhibitor with people, a concentration of 100 nanomoles per dm cubed would be the most appropriate to use. Do these data support the student's conclusion? Give reasons for your answer. So looking at the data, do you think that a concentration of, 100 nanomoles per dm cubed is the most effective concentration? You can give reasons for and give reasons against, but you need to use the data. Don't give me an answer without any data. The final question. At the start of their investigation, the scientists made a solution of kinesin inhibitor with a concentration of 10,000 nanomoles per dm cubed. They use this to make the other concentrations by a series of dilutions with water. Describe how they made 100 centimetres cubed of 100 nanomole per dm cubed solution of kinesin inhibitor. So you need to tell me how much of that original solution they would have added in and how much of the water they would have added in to dilute it. Okay, they told you that it's made a series of dilutions with water. On the next slide, you're going to see the answers. So pause now if you haven't answered those. Look at the PowerPoint in Edmodo if you've missed any questions, or you can just rewind me. And now let's have a look. So the first question was looking at how the monopolar mitotic spindle will prevent mitosis. No separation of chromatids, chromosomes or centromeres. So the chromatids or chromosomes all go to one pole, or they're not poled to opposite poles. And this doubles the chromosome number in cell, or one of the daughter cell gets all the chromosomes and the other gets none. Okay. Next question was the big one. Okay, it was looking at that data set. Do these data support the conclusion that 100 nanomoles was the best or the most suitable for the treatment? Okay, so you should have had yes and no. Okay, no, it's not suitable because at 100 nanomoles there was only 93% of the cells showing that monopolar mitotic spindle. So some of those cells, bearing in mind that they're cancer cells, can still divide. This means that the cancer isn't destroyed, or it might continue to grow. You could suggest another concentration that might be better. So you might suggest that they need some more trials between 100, where there was 93% monopolar mitotic spindle, and 1,000 nanomoles, where there was 100%. You could suggest that again. Okay, there's maximum 4 marks for this at any of these 4 points. You could have also said that this research was done in a culture or a plate of cells, so we don't know how this kinesin inhibitor affects people. Okay, don't say though that it's not tested on humans. You needed to say that it's done in a culture, so it's done in bacteria first, and we don't know how it's going to affect individuals. Now you could have said yes, that 100 nanomoles is fine, because any further increase above 100 nanomoles has a little increase in the percentage of cells not undividing or undergoing mitosis. Or you could have said that at 100 nanomoles, most of the cancer cells are unable to divide. You could have also suggested that above 100 nanomoles, or at higher concentrations over here, there might be harmful side effects that we don't know. You could have also mentioned that any higher concentrations are more expensive, and that above 100 might have more effect on rapidly dividing cancer cells. Now the